Welcome, curious minds, to the deep dive. Have you ever truly paused to consider the unseen worlds tirelessly working behind the scenes, creating so many of the things we rely on every single day? I'm talking about the microscopic maestros, the tiny powerhouses we call microbes, and the incredible transformative power of fermentation. Think about it. From life-saving medicines and groundbreaking gene therapies to the clean biofuels powering our future, even the everyday food and drinks we enjoy, so much of it hinges on these invisible biological processes. But here's the compelling thought, the subtle hum beneath all that innovation. While fermentation is undeniably powerful and revolutionary, unlocking countless possibilities, it also comes with inherent and sometimes surprisingly hidden risks. This leads us to a truly critical question, one that shapes how we interact with this technology. How do we ensure this revolutionary process, which impacts so many facets of our lives, is truly safe for everyone? Today, we're plunging headfirst into that very question. Our deep dive is all about biosafety and fermentation technology, a crucial yet often overlooked aspect of this vital field. And we're drawing our insights directly from the comprehensive and incredibly illuminating analysis by Dr. Rama Yusmana from the Faculty of Engineering Technology at Tun Hussein on University of Malaysia, or UTHM. As we unpack this, you might be surprised by the sheer variety of hazards involved, extending far beyond what you typically imagine. We'll understand exactly how we might accidentally encounter them, and then, perhaps most fascinatingly, explore the elaborate, multi-layered systems put in place to keep us safe, all illuminated with vivid, real-world examples.
That's a perfect setup. And Dr. Yuzana's work, it really redefines biosafety. It's not just a narrow concern limited to the confines of a laboratory bench, dealing with a spilled beaker. Instead, biosafety is presented as a critical foundational element for broader public health and environmental protection. When we talk of biohazards in this context, Dr. Yusvana offers a clear foundational definition. These are biological agents or conditions that pose a distinct threat not just to humans but also to animals or to the wider environment. It's an incredibly comprehensive view, encompassing a vast array of potential dangers. What's particularly insightful here is how Dr. Yusvana broadens the very scope of biosafety beyond just preventing harm. It's about a dual importance. Yes, it's absolutely vital for ensuring the safety of workers directly involved in the process, the general public who might encounter products, and the environment from direct exposure to harmful agents. But equally, and perhaps less intuitively, it's about preventing contamination of the very products being created and the facilities themselves. Think of it. If a pharmaceutical batch is contaminated, it's not only a safety risk, but also a massive economic loss and a public health concern if it's distributed. This aspect of internal protection is incredibly important for industries heavily reliant on fermentation, where product purity, integrity, and consistency are, well, non-negotiable. So if we connect this to the bigger picture, biosafety ensures both external protection for people and the planet, and internal protection for the integrity of the process and its outputs. To truly understand how this is achieved, we first need to dive into the first major pillar, understanding the diverse and sometimes surprising types of biohazards we encounter in fermentation.
All right, that sets the stage. Now, let's pull back the curtain on the actual threats. When most of us hear biohazard, our minds race to Hollywood thrillers and dangerous viruses. But Dr. Yusvana's deep dive reveals a much more nuanced and often surprising spectrum of risks right within the world of fermentation itself. It's not just the obvious bad guys we need to worry about, is it? Let's start with the biological agents themselves, because they are, after all, at the very core of fermentation.
Indeed. The most intuitive biohazards are, of course, the pathogenic microorganisms, things like bacteria, viruses, and fungi that can cause disease in living organisms. But it's rarely as simple as good microbe versus bad microbe. Consider E. coli, for instance. This bacterium is a true workhorse in many fermentation labs. It's widely used in genetic engineering and protein production to create everything from recombinant insulin, which saves millions of lives daily, to other vital pharmaceuticals. However, here's where the unexpected twist comes in. While most E. coli strains are perfectly harmless and even beneficial to human gut health, some, like E. coli 0157, are notorious. This particular strain can cause severe food poisoning, leading to debilitating cramps, bloody diarrhea, and in severe cases, kidney failure. This highlights a crucial point that Dr. Yusvana emphasizes. Even a common, industrially useful organism can have dangerous variants, making precise strain identification, meticulous handling, and rigorous containment absolutely critical to prevent outbreaks.
Wow, okay, so even something really common in labs can have a dangerous side.
Exactly. And then we have adenoviruses. These are truly fascinating in their dual nature. In the world of biotechnology, they're critically important as viral vectors in gene therapy. They are modified to act as delivery vehicles, transporting therapeutic genes into human cells to treat a wide range of genetic disorders, from cystic fibrosis to certain cancers. This is revolutionary medicine. But precisely because they're viruses, even when engineered for beneficial purposes, their handling and containment must be incredibly careful, almost to a fault. The potential for unintended spread or infection, even with attenuated or modified versions, is a constant, paramount consideration that dictates every safety protocol around them.
So you're using a virus to deliver medicine, but you have to be super careful it doesn't act like, well, a virus.
Precisely. Let's not forget fungi, like Aspergillus niger. This fungus is another industrial workhorse, utilized extensively for producing essential enzymes and organic acids, most notably citric acid, which is found in countless food products and beverages. Yet here's the critical caveat, a stark reminder of the complexities. Certain strains of Aspergillus have the potential to produce harmful compounds known as mycotoxins. These are secondary metabolites that can be toxic or carcinogenic. Aflatoxin, a particularly potent mycotoxin we'll discuss further, is a prime example. So while Aspergillus niger is industrially beneficial, stringent biosafety measures are absolutely essential to prevent any accidental release of mycotoxin-producing strains or contamination of products with these toxins, which could have serious long-term health implications for consumers.
Mycotoxins, that sounds pretty nasty.
They can be, yes. Moving beyond naturally occurring pathogens, Dr. Yuzvana also highlights genetically modified organisms, or GMOs, as a significant biological hazard, truly a double-edged sword in the world of fermentation. These organisms, whether they are bacteria, yeast, or fungi, are meticulously engineered to enhance the production of desired products. For example, recombinant yeast, meaning yeast that has been genetically engineered, is a cornerstone in producing pharmaceuticals like insulin, making life-saving drugs more accessible and affordable, or in creating new microbes for biofuels, driving sustainable energy solutions. The promise of GMOs in fermentation is immense, offering unprecedented control and efficiency in biological production. However, this is where it gets truly thought-provoking, because Dr. Yuzvana issues a clear warning that compels us to think deeply about responsible innovation. There is the potential for the unintended spread of modified genes to wild populations. Imagine a gene engineered for a specific industrial purpose, perhaps for increased enzyme production, somehow making its way out of the contained environment and into the natural ecosystem, altering wild species in unforeseen ways. Even more concerning is the possibility, though carefully managed, of creating new pathogens with novel, unforeseen characteristics. This isn't about demonizing the incredible power of genetic engineering. Rather, it's about controlling that innovation responsibly, ensuring that our advancements don't lead to unintended biological consequences for our environment or public health. It's a constant balancing act between scientific progress and diligent oversight.
That's a really crucial point. Innovation is great, but you have to think about the what-ifs.
Absolutely. And finally, within the realm of biological agents, we must consider microbial toxins. These are invisible threats, often potent. They're toxic substances produced by some bacteria and fungi, frequently as a defense mechanism to outcompete other microbes in their natural environment. A chilling and yet fascinating example is botulinum toxin, produced by the bacterium Clostridium botulinum. This is a neurotoxin of extreme potency, considered one of the most toxic substances known to science. Even tiny amounts can be lethal, leading to paralysis and respiratory failure. Yet remarkably, this same toxin is precisely controlled and used in minute, incredibly controlled quantities for medical purposes, most famously as Botox, to treat muscle spasms and cosmetic wrinkles. This vividly illustrates the precise, meticulous control and understanding needed when working with such dangerous compounds, transforming something deadly into something therapeutic through careful application.
Wow, the Botox toxin. I never thought of that as a biohazard in this context, but it makes perfect sense. The control has to be absolute.
Exactly. And on the other end of the spectrum, you have aflatoxin, produced by certain Aspergillus species, which we briefly mentioned. This is highly carcinogenic, meaning it can cause cancer, and poses a significant risk when it contaminates food supplies, especially grains and nuts. It's a critical public health concern, particularly in agricultural and food industries worldwide, demanding rigorous testing and prevention protocols.
That truly paints a complex picture, extending far beyond just the organisms themselves. It sounds like a complex web of interactions. But Dr. Yuzvana dives deeper, showing us that the risks aren't exclusively biological, are they? There are often overlooked chemical and even physical hazards right within the world of fermentation.
Indeed, the complexity grows when we consider the entire ecosystem of a fermentation process, which extends far beyond the microbes. The process itself involves a host of chemical components, many of which can be hazardous. For instance, antibiotics like penicillin or streptomycin are often added to the fermentation media to maintain culture purity, preventing contamination from unwanted bacteria or fungi. This is crucial for product consistency. However, the hidden danger here lies in improper disposal of waste. If antibiotic-laden waste enters the environment untreated, it can inadvertently contribute to the development and spread of antibiotic-resistant bacteria, which is a major escalating global health threat. Beyond specific additives, many of the general chemicals used in various stages of fermentation, from pH adjusters to cleaning agents, can be hazardous due to their inherent flammability, toxicity, or potential to cause respiratory issues simply by their presence in the workspace. Careful handling and storage are paramount.
So the stuff you add to keep things clean could actually cause a different problem down the line. That's ironic.
It is, in a way. And then there are the toxic byproducts and metabolites that the microbes themselves produce as part of their metabolic processes. Consider lactic acid produced by certain bacteria in many industrial fermentations, like those for dairy products or bioplastics. While essential for these processes, its accumulation can lead to harmful acidic conditions in the culture medium, affecting the microbe's health and potentially corroding equipment. If not properly managed, this acidic waste can also pose an environmental risk. Another interesting example is fusel alcohols, such as isoamyl alcohol, which yeast might produce as a byproduct during ethanol fermentation. While part of the flavor profile in some beverages in higher concentrations, these fusel alcohols can be toxic if consumed, adding another layer of complexity to product safety and quality control, especially in the beverage industry. It's a reminder that even the desired output can have undesired co-products.
And it's not just the ingredients or what the microbes make, it's even the equipment we use, isn't it? The very tools of fermentation, essential for the process, can pose significant, often overlooked risks. How do these physical elements contribute to the biosafety challenge?
That's a crucial insight, and Dr. Yuzvana meticulously details it. The laboratory and industrial equipment itself can be a source of significant physical hazards that can exacerbate or create biohazard risks. Autoclaves, for instance, are absolutely essential for sterilizing equipment and media, ensuring a sterile environment critical for preventing contamination in fermentation. But they operate at very high temperatures, often exceeding 121 degrees Celsius, and intense pressures, typically around 15 pounds per square inch above atmospheric pressure. This means they inherently pose severe risks of burns or even explosions if not maintained properly or operated according to strict safety protocols. Imagine the destructive power if a seal fails under that pressure.
Right, you hear about autoclave accidents, high pressure and heat. Definitely dangerous.
Definitely. Centrifuges are another example. These machines spin at incredibly high speeds, often tens of thousands of revolutions per minute, to separate cellular components from liquid media. Mechanical failures, such as an unbalanced rotor or material fatigue, can lead to catastrophic breakage. This not only risks releasing biohazards if samples containing infectious agents are being spun, but it can also send flying fragments of metal or glass into the lab, posing severe physical injury risks to anyone nearby. It's a reminder that mechanical integrity is intrinsically linked to biological safety.
Flying metal shards. Yeah, that doesn't sound good in a lab setting.
Not at all. And then there are the bioreactors themselves, which are the core vessels for fermentation. These can range from small lab-scale flasks to massive industrial tanks spanning multiple stories. These large-scale systems carry their own set of critical risks, including leaks of vast volumes of media containing active cultures, spills during transfer or sampling, and pressure-related accidents due to gas buildup or sudden changes in temperature. Any of these can lead to widespread contamination, exposure to hazardous chemicals or microbes, or even physical injury from explosive decompression or scalding liquids. Each component of the system, from pumps to valves, must be meticulously designed and maintained for integrity.
So the heart of the operation, the bioreactor, is also a potential point of failure.
It can be, absolutely. Beyond specific equipment, the environmental conditions within a laboratory or large-scale facility can also contribute significantly to physical hazards. Exposure to extreme cold, for instance, is common when handling cryogenic samples materials stored at extremely low freezing temperatures, typically in liquid nitrogen, to preserve microbial cultures or biological materials. This poses risks of frostbite or hypothermia to personnel. Conversely, high temperatures in certain process areas, perhaps from heat-generating fermentation, can lead to burns or heat exhaustion. Furthermore, poor ventilation is a significant and often insidious concern. Inadequate airflow can lead to the dangerous accumulation of hazardous substances in the workspace, whether they are chemical vapors from solvents or airborne microbial particles from aerosol-generating procedures. This directly impacts personnel health over time, potentially leading to respiratory illnesses or systemic toxic effects. It's a silent threat that requires constant monitoring.
So what does this all mean? It seems like these hazards aren't just isolated incidents that pop up randomly. They can stem from every single stage of the fermentation process, from the very beginning with the raw materials to the very end with waste. It's a continuous chain of potential exposure points, almost like a complex domino effect waiting to happen if not properly managed.
Precisely. Dr. Yuzvana meticulously breaks down where these dangers lurk throughout the entire fermentation life cycle, emphasizing that safety begins long before the microbes even enter the bioreactor. It starts right with the raw materials. Substrates and nutrients, which are essential for microbial growth, can become contaminated with unwanted microorganisms or chemical impurities during their handling, storage, or transportation. These common contaminants, whether they are wild bacteria, molds, or even residual pesticides, can outcompete or harm the desired culture, leading to reduced yields, altered product quality, or even outright batch failure. Similarly, non-sterile water, often used as a solvent or for rinsing, can introduce a wide range of unwanted elements, including other microbes, organic matter, and chemical impurities, all of which negatively affect the delicate balance of the fermentation process and the ultimate purity of the product. The quality control here is the first line of defense.
So even the water you use needs to be perfect.
Essentially, yes. Then we move to the inoculum, the crucial starter culture itself. Pathogenic strains may be intentionally used for very specific research or production purposes, such as in the development of attenuated vaccines or as biocontrol agents, which are living organisms used to control pests or diseases. However, their very nature means they pose significant biohazard risks, including direct infection of laboratory personnel, widespread contamination of the facility if containment fails, and potential environmental release, which could have ecological consequences. Even with non-pathogenic strains, contamination can occur during preparation through various means, like airborne particles, inadvertent contact with unsterilized equipment, or contact with non-sterile surfaces. This seemingly minor contamination can lead to reduced fermentation efficiency, altered product characteristics, and in worst-case scenarios, the complete failure of the fermentation process, leading to significant economic losses.
So getting the starter culture right and keeping it clean is absolutely critical.
It's fundamental. Process conditions themselves are another critical source of hazard that require constant vigilance. Suboptimal sterilization, for example, is a common culprit. If equipment, media, or containers are not thoroughly sterilized, it can lead to the survival and proliferation of unwanted microorganisms in the fermentation process. This not only compromises product quality and purity, but also poses safety risks, as these contaminating microbes could be harmful. Ensuring thorough and effective sterilization of all equipment, media, and surfaces is therefore absolutely essential to prevent these biohazard issues. Also, inadequate containment and handling practices during the fermentation process are major pathways for risk. If hazardous microorganisms or chemicals are not properly contained or handled, they can escape the controlled environment, posing direct risks to personnel through splashes or aerosols, contaminating the facility, and potentially leading to environmental release. Implementing strict containment protocols, such as closed systems and biosafety cabinets, and rigorous handling techniques is crucial to maintaining biosafety throughout fermentation operations.
Containment seems like a really recurring theme here, keeping things where they belong.
It's one of the core principles. Finally, we arrive at waste, and effluence the end products of the process that must be managed carefully. Improper disposal of biological waste, such as spent microbial cultures, contaminated media, or used PPE, can lead to widespread environmental contamination and the proliferation of hazardous microorganisms. This creates significant public health risks, as these pathogens could find their way into water sources, soil, or even agricultural products. It's not just about what's inside the lab, but what ultimately leaves it. Ensuring that all waste is appropriately decontaminated, often through autoclaving or chemical treatment, and disposed of according to stringent biosafety guidelines, is absolutely crucial to prevent such hazards. Furthermore, the release of untreated effluents, the liquid waste streams from fermentation processes, can introduce harmful microorganisms and toxic byproducts into water systems, posing severe ecological and health risks to aquatic life and downstream communities. This makes implementing effective treatment and containment systems for effluents, like filtration and chemical neutralization, essential to safeguard both the environment and public health.
Wow, okay. So knowing what the hazards are and where they originate throughout the entire process is truly crucial. But Dr. Chizana then shifts our focus to how these hazards can actually reach us. Understanding these routes of exposure is absolutely key to building proper defenses because you can't protect against a threat if you don't know its pathways. How do these insidious agents typically make their way into our systems?
That's a foundational question for any safety strategy. There are four primary routes through which these biohazards can enter our system, and understanding each one is vital for effective prevention. First, and perhaps most prevalent in any microbiology or fermentation lab setting, is inhalation. Many fermentation processes, especially those involving agitation, stirring, or transfer of liquids, can generate fine mist or aerosols. These invisible particles can contain harmful microorganisms or chemical agents, posing direct inhalation risks to laboratory personnel. Similarly, when handling dried microbial cultures, which are often used as inocula or for storage, dust particles can become airborne. If inhaled, these can lead to respiratory issues or even infections. The primary prevention strategies here are robust, using completely closed systems wherever possible to prevent aerosol generation, ensuring proper ventilation in all work areas with controlled airflow, and consistently using personal protective equipment, PPE, like N95 respirators or surgical masks where appropriate. Additionally, working within certified biosafety cabinets, which capture and filter airborne particles, and implementing stringent cleaning procedures helps minimize aerosol and dust exposure in the workspace.
Aerosols. Those invisible mists sound like a major challenge to control.
They are. The second significant route is ingestion, which often happens through accidental swallowing. This can occur when laboratory personnel handle biohazardous substances without following proper hygiene practices, such as eating, drinking, or applying cosmetics in the lab, or even just unconsciously touching their face with contaminated hands. There's also the risk, particularly in food or beverage fermentation, of consuming food or water contaminated with hazardous agents from the fermentation process itself, perhaps through cross-contamination in shared facilities. To prevent this, strict adherence to no eating or drinking policies in the laboratory is paramount, alongside rigorous, regular hand washing, especially after handling materials and before leaving the lab. Proper storage of all food and beverages away from lab areas, combined with comprehensive training for lab personnel in contamination prevention, significantly reduces this critical risk.
Basic hygiene, but absolutely critical in that kind of environment.
Incredibly critical. Third, we have skin contact. Direct handling of contaminated materials, such as microbial cultures, fermented media, or concentrated chemical reagents, can result in skin exposure to harmful agents. This can lead to localized infections if the skin is broken or even intact, or chemical burns and irritation. Think of a splash while pouring a liquid or brushing against a contaminated surface. Unforeseen spills and splashes, which are always a possibility during dynamic processes like fermentation, can also expose large areas of the skin to biohazardous or toxic substances, posing significant health hazards. The prevention here is clear and direct. Wearing appropriate PPE like impervious gloves, which are chosen based on the chemicals being handled, and lab coats is absolutely essential to create a barrier against these risks. Additionally, prompt cleanup of spills with proper disinfectants and using physical barriers like splash guards on equipment can significantly minimize the risk of accidental skin exposure.
Gloves and lab coats, the classic lab gear, but for very good reason.
Essential barriers. Finally, and perhaps most acutely dangerous due to its direct entry into the bloodstream, is injection. This primarily refers to accidental needle sticks or cuts, which can occur while handling sharp instruments like needles, scalpels, broken glass, or even pipette tips that might have sharp edges. Such punctures create a direct pathway for hazardous microorganisms or chemicals to enter the body, potentially leading to severe infections or injuries that bypass the body's natural external defenses. Beyond needles, improper handling of other sharps, such as broken glassware or sharp tools used for maintenance, significantly increases the likelihood of accidental punctures or lacerations, posing considerable biohazard exposure risks. The prevention strategies are critical and non-negotiable. This includes using safety-engineered needles that retract or shield themselves after use, ensuring proper disposal of all sharps immediately after use into designated puncture-proof containers, and providing comprehensive hands-on training on safe handling practices for all sharps. Strict adherence to standard operating procedures or SOPs for all sharp-related tasks is absolutely essential to minimize this high-risk route of exposure.
Needle sticks. Yeah, that's a scary one. Direct line in.
Extremely high risk. So we've identified the threats, and we know exactly how they can get to us, the insidious ways they can enter our system. Now comes the truly critical part. How do we manage this risk? How do we take all this knowledge of potential danger and transform it into a practical, actionable plan for safety? This is where Dr. Yuzwana's framework for risk assessment and management strategies truly shines, essentially transforming potential disaster into meticulously controlled innovation.
Right, the action plan. How do you build the defenses?
This is indeed the core of effective biosafety. It's about being proactive, not just reactive. It all starts with robust risk assessment, which Dr. Yuzvana describes as the detective work, the meticulous investigation that precedes any action. The very first step is identifying potential hazards. This means recognizing all biological, chemical, and physical risks associated with every single stage of the fermentation process, from the incoming raw materials through the act of fermentation to the outgoing waste. It's about anticipating threats, asking, what pathogenic microorganisms might be present? What toxic chemicals are involved? What equipment malfunctions could lead to exposure? A thorough hazard analysis at each and every process stage is absolutely crucial for this anticipation and subsequent mitigation. This isn't a quick checklist, it's a deep dive into every step.
So you map out everything that could go wrong, basically.
Pretty much, yes. Once hazards are identified, the next step is evaluating exposure levels and consequences. This involves assessing how and to what extent personnel might actually come into contact with the identified hazards, considering factors such as the frequency of exposure, is it a daily task or a rare maintenance event, and the duration of that contact. Simultaneously, you analyze the potential consequences of that exposure, including both the immediate and long-term health impacts on personnel and any potential disruptions to the fermentation process or product quality. This comprehensive evaluation aids in truly understanding the overall severity and likelihood of the risks involved. Finally, you use this information to determine precise risk levels by combining the likelihood of exposure with the severity of the consequences. This allows you to categorize the overall risk as low, medium, or high, which then directly informs the development and implementation of appropriate, tailored safety measures and protocols to effectively manage and minimize the identified risks. It's a precise calibration process.
Identify, evaluate, categorize. Makes sense. Then what?
Then, once risks are meticulously assessed, we move into the actual risk management strategies, essentially, the comprehensive defense plan. These strategies are typically layered, following a hierarchy of controls, with the strongest and most effective controls prioritized. The first and most impactful are engineering controls. These involve physical modifications to the workspace or equipment itself, specifically designed to contain hazardous agents and prevent their release or personnel exposure at the source. Examples are numerous and vital. Using completely closed systems for fermentation, where the microbial culture never touches the outside environment, minimizing any chance of escape. Another critical engineering control is the use of biosafety cabinets, which create a protected workspace with filtered airflow to contain aerosols and prevent inhalation exposure. Proper ventilation systems throughout the facility, equipped with high-efficiency particulate air or HEPA filters. These are essentially sophisticated air purifiers designed to trap even the tiniest microscopic airborne particles, are also vital engineering controls, ensuring the removal of harmful aerosols and maintaining safe, clean air quality in the laboratory or facility. These controls remove the hazard before it ever reaches a person.
Building safety right into the equipment and the building itself. That seems like the smartest first step.
It's considered the most effective layer, yes. Next come administrative controls. These are procedurals focusing on how work is done safely, establishing the rules of engagement. This includes developing and rigorously enforcing standard operating procedures, or SOPs, that clearly outline safe practices and protocols for handling all biohazards and chemicals. These aren't just guidelines, they are mandatory step-by-step instructions for every task involving risk. Equally vital is ensuring regular, comprehensive training sessions and certification programs for all personnel. This isn't a one-time onboarding, it's an ongoing process that ensures everyone working in the facility is not only knowledgeable about these complex procedures, but also consistently competent in their implementation. This builds a proactive culture of safety from the ground up, where every individual understands their role in maintaining biosafety.
So engineering fixes the environment, admin fixes the process, and the people.
That's a good way to put it. Finally, we have personal protective equipment, or PPE. While often the most visible, PPE, like gloves, masks, and goggles, provides an essential final layer of defense against exposure to hazardous agents by creating a physical barrier to protect the skin, eyes, and respiratory system. The emphasis here, as Dr. Uzana notes, is not just on having PPE available, but on it being worn consistently, correctly, and appropriately for the specific task and hazard. It's a critical barrier, but it's most effective when used in conjunction with robust engineering and administrative controls. It's the last line of defense, a personal shield, when all other barriers have been put in place.
The gear is important, but it's the last resort, not the first thought.
Ideally, yes. And this all feeds into what many in the science world know as biosafety levels, or BSLs. It's a tiered system, like layers of an onion, designed to match the level of containment to the specific level of risk involved. This tiered approach is fascinating because it allows for such a precise calibration of safety measures. It's truly a testament to how thoughtfully these systems are designed.
Right, the BSLs, I've heard of those. Can you walk us through these levels? It sounds like a really structured way to handle different degrees of danger.
That's an excellent analogy. Layers of an onion, or perhaps a series of fortresses, each more robust than the last. The biosafety levels, or BSLs, provide a globally standardized framework for the safe handling of biological materials, escalating in stringency as the risk associated with the biological agent increases. We start with BSL-1. This is essentially the basic laboratory, designed for work with well-characterized agents that are not known to consistently cause disease in healthy adult humans. They pose a minimal potential hazard to laboratory personnel and the environment. Think of common, non-pathogenic strains of E. coli, often used in educational settings, or baker's yeast. For BSL-1, basic lab safety practices are sufficient. This includes routine hand washing, a strict no eating or drinking policy in the lab area, and the use of basic personal protective equipment like lab coats and gloves to prevent contamination. Procedures also cover proper disposal of routine laboratory waste in designated containers and regular disinfection of work surfaces to maintain a safe and clean working environment. It's all about good basic microbiological practice, the foundational habits of a safe lab.
So BSL-1 is kind of like your standard high school or college biology lab, maybe?
Roughly, yes, in terms of the types of agents and basic precautions. Stepping up, BSL-2 is designed for agents posing moderate hazards to personnel and the environment. These agents can cause human disease, but typically through accidental ingestion, inhalation of aerosols, or direct skin exposure rather than casual contact. Examples include common pathogens like Staphylococcus aureus, which causes staph infections, or certain strains of Salmonella, known for food poisoning. At this level, you see a significant enhancement of lab practices and equipment. This includes strict adherence to detailed standard operating procedures, SOPs, for every task. Mandatory use of PPE becomes more rigorous, encompassing gloves, lab coats, and face protection, like safety glasses or face shields, especially during procedures that might generate splashes or aerosols. Laboratories must also have ready access to Class II biosafety cabinets for any procedures that generate aerosols or involve infectious materials, providing a primary barrier. Additional safety measures include the use of autoclaves for decontaminating all infectious waste before disposal, and proper documented training for all personnel to ensure they can handle these moderate biohazards safely and competently.
Okay, so BSL-2 ups the ante quite a bit. More rules, more gear, special equipment like those cabinets.
Exactly. Then there's BSL-3. This level is reserved for work with indigenous or exotic agents that may cause serious or potentially lethal diseases, specifically through inhalation. These microorganisms necessitate stringent biosafety measures to protect laboratory personnel and prevent any environmental release. Think of pathogens such as Mycobacterium tuberculosis, the bacterium responsible for tuberculosis, or the SARS-CoV-2 virus, which caused the COVID-19 pandemic. The high risk associated with these airborne agents transforms the lab into a fortress. Laboratory practices at BSL-3 include strictly restricted access, allowing only authorized and fully trained personnel. There are rigorous decontamination protocols for all equipment and surfaces, ensuring no pathogen leaves the contained area. All work involving these agents must be conducted within certified biosafety cabinets, typically Class II or Class III, which provide a high level of containment by filtering air and creating physical barriers. The laboratory facility itself must have specialized ventilation systems with HEPA filtration and directional airflow, meaning air flows into the lab but not out, to ensure contaminated air cannot escape. Furthermore, personnel are required to undergo specific, intensive training in handling pathogenic and potentially lethal agents, and must adhere to stringent PPE requirements, which often include respiratory protection like N95 masks or powered air-purifying respirators.
BSL-3 sounds serious. Restricted access, special air systems, dealing with things that can really harm you through the air.
Very serious, yes. Finally, we reach BSL-4. This is the absolute pinnacle of containment, the highest level of biosafety. It's reserved for work with dangerous and exotic agents that pose a high risk of life-threatening disease, often lacking available vaccines or treatments. Classic examples include the Ebola and Marburg viruses, notorious for their high mortality rates and the ability to cause severe diseases through aerosol transmission. At BSL-4, maximum containment practices are implemented, creating an almost completely isolated environment. This means personnel wear full-body, air-supplied positive pressure suits like astronauts in a hostile environment to protect them from any exposure, breathing filtered air from an external source. All work is conducted either within Class 3 biosafety cabinets, which are completely sealed glove boxes, or in these fully contained ventilated suits within specially designed containment facilities. The laboratories must have dedicated air supply and exhaust systems that are completely independent of other building systems, rigorous decontamination procedures for all materials exiting the lab, including shower-out procedures for personnel, and extensive ongoing training for personnel in emergency protocols and disaster response. It is, quite literally, a true fortress of safety, built to contain the most dangerous known pathogens.